Okay, I make that the hour, so I think we'll get started. My name is Chris Thorne. I am Senior Manager for Field Marketing here at Twist Bioscience. A very warm welcome to you, either this morning or this afternoon or wherever you are in the world. Before we begin, just a little bit of housekeeping. All lines will be muted during the webinar, so you'll notice that your microphone is muted. But we will be taking questions at the end. If I can ask you to submit your questions in the Q&A box, which you'll find at the bottom of the Zoom interface, rather than the chat box. And this way we'll be able to deal with those appropriately. Following the presentation and the Q&A, if you do have a minute at the end, we will forward you on to a brief survey, and this will just be allow you to give us a little bit of feedback and help us improve on these things. Okay, so with that done, I'm now going to be moving on. So it gives me great pleasure to introduce Dr. Ulrich Elling. Dr. Elling runs an academic lab at the IMBA in Vienna, Austria, where his group is focused on cell identity transitions, as well as the development of genetic screening tools. His recent work has focused on improvements of CRISPR-Cas9 as a genetic screening tool. Two examples being CRISPR-UMI, a single-cell tracing method for CRISPR screening, and also CRISPR-Switch, a tight and rapidly inducible control of sgRNA activity. So we're very fortunate to be joined by Dr. Elling today to talk about his lab's recent publication in Nature Methods, detailing the development of a new multi-layered sgRNA score that markedly outperforms all previous prediction tools. So Ulrich, I'm very much looking forward to this, and without further ado, I'm going to hand over to you.
Yes, so hello everybody, I hope you can see me. I am talking to you from Vienna today, and welcome to everybody. Good morning, good evening, wherever you are. It's good to meet you in these special days. And because it is special days and we will be doing this online, I reserved a little corner in the top right where you can basically put me and your chat interfaces. I will not be using this space for the data that I will present. Now, there we go. So we will be talking about screening, and I just want to point out as a starter that a genetic screen using any mutagenesis method is really a multi-step process. In the case of CRISPR screening, it starts with guide RNA design, as we will talk about today, and then guide RNA synthesis, as Twist is specialized in, but then also, of course, the cloning and the cell culture work, followed up by PCR and next generation sequencing, where we will get some pixels from a flow cell that eventually we want to translate into human biology. Now, this is a very non-intuitive process. It's multi-step, and it requires p-values that go well beyond our classical 0.05 that we are used to in biological experiments. If we look into the details of how such genetic screens are done, then it typically starts with a cell line where we would put in Cas9. We actually do this in a clonal way because we find this to work much better. Subsequently, we transfect the cell line or transduce the cell line with guide RNAs, select for the presence thereof. And then if we hit, for example, a tumor suppressor, that guide RNA would enrich in the population, while essential genes targeted would result in the death of the cell and therefore a depletion. And we compare the before and after by next generation sequencing, but abundance of guide RNAs is not the only way that you can assay your screen. I'm sure you're all aware of the many other methods such as crop-seq, morphology-based assays, of course, editing outcomes, so studying the edit itself, and so on and so forth. That all can be an output of CRISPR screens. Now, if you talk about genome editing, when we refer to CRISPR, we are actually making a little bit of a jump, because we are not editing the genome. We're just destroying the genome in a controlled manner, and then we hope for some sort of mutation to occur. And this indirect measurement, measuring the presence of a guide as opposed to the presence of a mutation, actually results in a sort of ambiguity in the results of a screen. Maybe as an introduction, there's really, if you talk about guide RNA abundance, the classical way of doing CRISPR screens, there's two ways that people do this. So in the end, you measure the abundance of the guide RNA in your next generation sequencing reads. And so the guide RNA can either be positively or negatively selected for. A positive selection is the classical experiment of finding a needle in a haystack. And this sounds challenging in the first moment, but if you think about just burning away the haystack, all you're left with is the needle. So in the end, positive selection is actually not so difficult. Negative selection, however, requires you to identify the type of grass that is missing from your haystack. And that really requires quantification of all the different cells in your screen and so on, and is much more challenging. However, it is more rewarding, and so most people, or there's a more broad application for this, and so most people are actually focusing on a negative selection. Here's some experiments that were done in the Zuber lab here in Vienna at the IMP, and you can see that so every dot that you're seeing in these graphs is a different gene, and you can see that they deviate quite far from the zero. The red ones are the core essential genes. So you can see that the median core essential gene is actually depleting by a log fold of about -4 to -5, which represents a depletion of 95% from the population. And this depletion that we saw was very rewarding. It was very strong and also stronger than what other people had seen. But and the reason why we're so happy with this is because we knew that the magnitude of depletion actually in the core essentials here on the x-axis directly correlates to the signal-to-noise ratio within your screen. So the better you're depleting the genes that are essential, the better your signal-to-noise ratio is in the screen. We had previously, and that was mentioned already, contributed to a bioinformatical trick to improve the sensitivity of these screens with CRISPR-UMI. And in this plot, I should also mention, so every dot here is a genome-wide screen in another cell line. And basically, we mounted the big libraries here that are published in the public domain. Now here, again, in a more mathematical way, positive selection versus negative selection. And what you can see here is that to reach a 20-fold enrichment in a positive selection screen, you actually only need to generate less than 10% of loss of function alleles in your population. However, to reach a 20-fold depletion in a negative selection, you of course need to deplete from 100% down to 5%, so you need to have a 95% and above loss of function generation. So in other words, whether your negative selection screen works or not is really decided by this very last 10% on the scale, where you make the difference between a mediocre and a really good depletion for your genes. And that actually raised us to wonder, how can we reach such a strong depletion if what is expected to result in a depletion is a homozygous frame shift, shown here in this red bar in diploid cells. The top one is haploid cells. And you can see that with the expected ratio of 80% of loss of function alleles per allele in a diploid cell, you would actually only reach 1.5-fold depletion. So how come we saw a 20-fold depletion or 95% in our CRISPR screens? And so this is the questions I want to address. How can this unexpectedly strong dropout of core essentials be explained molecularly? And can we leverage these principles across the genome to make CRISPR screening more efficient? Now, our first hypothesis was that haploinsufficiency or dominant mutations contribute to this. And so we did a genome-wide screen in mouse embryonic stem cells and identified those genes that had a tendency to deplete stronger in diploid versus haploid cells. Because we reasoned that if a homozygous mutation is required, this should be more efficient in haploid cells, while if it's a dominant negative mutation, diploid cells should show stronger depletion because there's 2 chances to hit the genome. When we rescreened the same cells with a completely independent library, our UMI library, you can see that in this medium range of depletion, we actually had a significant confirmation. So the same genes tended to be more depleting in haploid or diploid, but the strong depletions were completely insignificant. This was noise, and we saw strong depletions both in haploid and diploid. So haploinsufficiency or dominant mutations don't seem to be explaining these strong dropouts. So the next idea was that it's because some of the guide RNAs only produce frame shifts. So we looked at guide RNAs that had either depleted very strongly, medium, or weak in our genome-wide screens, and asked the question whether these guide RNAs would generate different types of indels. And so Georg, the first author of this study, went through the heroic effort of analyzing guide RNAs in 2,400 data points, and to his disappointment, found that the weak, medium, and strong guides, strongly depleting guides, did not show any separation in their efficiency to produce frameshift mutations, neither as we measured them, nor as predicted by the two prediction algorithms in Delphi and Forecast. So we then looked into the details and pooled the three populations, and what we found, again, you can see in blue the in-frame mutations, so depletion here is up and insertion at the locus is down, and this would be 1 base depleted, 2 base, 3 base depleted, and so on. And you can see that the blue in-frame mutations are generated in all three groups, but in the weak, they remain, depleting guides, they remain, while in the medium and strong depleting guides, they do deplete just as well as the frameshift mutations. You can also see this in a kinetic way because Georg looked at this in different time points. And maybe quite illustrative here, the two genes NELF-B and IF3B actually deplete very similarly in the frameshift mutations. In fact, IF3B might even be a bit of a stronger depleter or a faster depleter. But because the in-frames remain for this gene and not for the other, this actually showed only a very weak overall depletion, while NELF showed a very strong depletion. So in a sort of provocative way, I would like to say that it's not the in-frame fraction, but whether they deplete those in-frames that defines guide RNA efficiency. Or in other words, gene lists after screens are sorted by gene targetability rather than by the biology that underlies them. And so with this in mind, we wanted to perform machine learning to get a deeper idea about what is going on with these protein levels. And we chose the Novartis tiled data set with 66,000 guide RNAs because that one was the only one that was performed in the improved guide RNA with an extended tracer, and at the same time neutral to any assumptions. If you look here across the big libraries from everybody, including ourselves, there's always a very strong bias in there already, and then you would be limiting the possibilities of finding new stuff. Now, mechanistically, there's multiple independent steps in editing. Everybody thinks about the Cas9, a bacterial enzyme, cutting DNA, but really it's not stopping there. After the cut of DNA, the mammalian DNA repair machinery comes in and generates these indels. And then again, mechanistically independent of that, there will be a transcriptional response, and such mutations can be spliced around, or alternative transcriptional start sites can rescue the situation. Subsequently, if the mutation remains in the mRNA, the ribosome will translate it. And if it's in frame, then it will terminate eventually. But if it's a frame shift, but if it's in frame, then a mutation will occur in a protein and will have some effect on protein function. Now, on the first level, we looked at cutting efficiency, and because we did our analysis on this improved tracer that shows higher efficiency with the AT swap and an extension in the repeat anti-repeat loop, we actually found a very different base pair tendency in this mask for the different positions compared to an earlier analysis with a shorter tracer. But let's focus on level 5 protein function, because I told you already that this is what separated our strong and weak guides from one another. And what we found here is that, of course, PFAM domain annotations are predictive in a machine learning algorithm for guide efficiency. But PFAM annotations are sort of a binary thing. And we found that if we analyze amino acid conservation, and more specifically amino acid conservation on a window of 7 amino acids across the cut site, then we get a better and more fine-grained predictor for guide RNA efficiency. However, and you can see this here, so on this example of FOXA1, where the PFAM domain targeted in this region is actually not so efficient because that would result in a depletion, which would go down. But depletion in this region is much better. And that is where the amino acid conservation is particularly high. You will also see, however, that there's this group of guide RNAs that didn't deplete at all, even though they have a very highly conserved amino acid code. And so we were wondering how we can identify or separate those ones. And what we noticed is that the type of amino acid that is encoded at the cut site, we're showing here a region of 5 amino acids around the cut site, is actually highly predictive of the effect of the efficiency of the guide, such that polar and charged amino acids are disfavored for good guides, while hydrophobic amino acids tend to be favored. The way we interpret this is that perturbations, so insertions or deletions at the core of the protein in the center, in the hydrophobic center, are much less tolerated than any changes in the surface. And you can see in this example of FOXA1 that now the amino acid identity put in a color code really clearly separates those guides that didn't work before as targeting unfavored amino acids. Now, we put all of this together with PFAM annotations, but also some aspects of gene structure. For example, we find that targeting bigger exons is better than small exons, and amino acid conservation and identity and so on, and we found that these individual parameters together are actually additive and add up to what we refer to as the Bioscore, a score that predicts the probability with which a mutation at a certain locus will result in a biological effect and thereby a phenotype. And we further combine this with our new score of guide RNA activity, as well as the frame shift prediction by Indelphi, which you can see has a fairly mild effect on large data sets, to what we refer to as the Vienna bioactivity CRISPR score, or VBC score for short. You can see that this Bioscore indeed is the strongest predictor for guide RNAs in this score together. And again, the 3 parameters are additive or synergistic and result in a better prediction. Now, to give you a visual idea what this looks like, here we put the Ruleset 2 by John Dönch. And you can see that, of course, Ruleset 2 predicts guide RNA cutting efficiency, but it has no parameters that will detect cold domains for editing, which we have integrated into the VBC score, and so we are getting a better predictive value. And I really want to highlight that good guide RNAs can in many cases be at the end of a gene. So this idea to target a gene early is really not a favorable strategy. And we've used it ourselves before, but it turns out not to be the way to go for good guides. Now, the images I just showed you you could argue are a circular argument because it's actually analyzed on the training set, but we next mounted all published screens from 7 different libraries and in hundreds of cell lines, and calculated the predictive value of our VBC score vis-a-vis all the other predictors that we had mounted. And you can see that in these data sets, by all 3 measures of predictability that we used, the VBC score actually outperformed all other predictors by a large margin. But the best way to prove this is, of course, a forward validation. So we called up Twist and asked them to synthesize a chip for us in super quick time because we were in revision. And they synthesized a chip for us with guides predicted by the Dönch score as well as our VBC score in one pool. We cloned them and screened them and found that indeed the VBC score performed much better than the classical or the commonly used density score. And most importantly, the VBC score performed really well across 4 bins of gene Bioscore. So here we calculate the median targetability or the median Bioscore for these genes and ask the question, and bin them by that. And you can see that if a gene is less easy to, is harder to target, then of course a classical screen will perform, classical score will perform worse, but our score seems stable. Now, what does that mean biologically? And what we argue is that as a take-home message, maybe that one aspect is that the current core essentialome is heavily biased for targetability. So genes that are easy to target, that everybody succeeds in targeting, will end up in the core essentialome because because they deplete everywhere. But other gene groups that are particularly hard to target will be missing. And maybe most dramatically, I would like to highlight here transcription factor activity, because of course transcription factors have large unstructured domains, so they are particularly hard to target, because if you target an unstructured domain, the in-frame mutations are not going to have any effect. The second point I want to make is that we are often blinded by the light when analyzing our screen data, because if you do an essentialome screen, we will have 2,000 or more genes significantly depleting. But on top of the list will be core essential genes with good targetability, or maybe some dominant mutations. But the new and interesting biology that you're seeking for might really be hidden in the lower range of depletion, simply because these genes are harder to target. And like I made the point before, these genes lists will be strongly sorted by targetability. Maybe also interesting for you to use this score. We've we generated a home page where you can go and look up for the screens. So we have for the guides that we predict. So we have different species in there, and you can just put your favorite gene name, like twist, for example, and go and design your guide RNAs. We also have a download section where you can download all guides in the genome, or 6 guides or so per gene, in order to generate big custom libraries. And I would, with the experience that we have in the lab, I would strongly recommend doing that, because it might take you some extra time to design your own library and with the best guides possible, and to clone that. But you will be rewarded by better data and a much more fun time during the validation stage of your screen data. So we just finished cloning genome-wide libraries for mouse and human. Again, Twist was very instrumental in helping us there. With this, I come to the end of this presentation. I'm very much hoping that there will be questions, and would love to answer some of them. Thank you very much.
Thank you, Ulrich. That was fantastic. So we do have some time for some questions now, and I can see them coming in by the Q&A box. So please continue to input your questions there. Just so you guys, our audience know, we also have first author on the paper, Georg Michlitz, who can join us for questions as well. So should we need to do so, there he is. Hi, Georg. So I'm going to begin with a quick question, just from me, actually, out of curiosity. So obviously we've seen, I think, a big improvement here in terms of our ability to predict and design high efficiency guides. Is the work done now, Ulrich, or do we continue to try to improve, or do you feel like this is the zenith? Where do we stand on this?
It's not at all done, unfortunately. So the scores that we used, some of them are actually quantitative. And what we can see is that so most of these other predictors reach a predictability of about 10%. We reach about 30%. But in an ideal scenario, you would be able to have an algorithm that predicts the very best guide RNA for every gene all the time, in which case it would be 100%. So there's still a long way to go. And, we have some ideas how this could be done. Thinking more about structure, putting in some some domain structures like alpha helices and so on, but also other stuff. No, it's still a long way to go. It's an improvement and it's a long way.
Yeah, brilliant. Okay, so really good question from Wei Li. So one possible explanation of strongly depleted sgRNAs is that they may have a high off-target activity. So did you consider off-target effects during the VBC score calculation?
Yeah, so that's definitely true. Thank you for that question. It is always a concern. Off-targets are always a concern. We do put in off-target rules, of course, quite stringent ones. And the guides that we predict, you can run any additional filter across them that you feel is adequate. Strictly speaking, off-target is, you just don't know it. So genome-wide, it'll be extremely hard to predict them. We have looked into this, and we don't see like higher off-target rate, predicted off-target rates for the guides we predict as opposed to other ones and so on. But that's also still prediction. One would have to go through probably thousands of guide RNAs and really do the experiment to formally rule it out completely.
Understood. So a couple of questions sort of related. So one asking about whether this algorithm can be applied to other species. And again, sort of similar, asking for non-mammalian species or even other nucleases. And I guess I have a sort of related question, which is, given that part of your algorithms related to conservation, will there be species where the strength of the algorithm starts to become less strong, or would you expect the score to apply across species?
Yeah, so conservation is one predictor for us to because evolution did the experiment to make mutations. So using conservation is one way of guessing whether a certain protein domain is of importance. We have many sequences in now, of course, so we can calculate conservation starting from multiple different species. In our homepage, we provide 5 species at the moment, so predictions for 5 species. We want to add up much more to that, but that's still not done. Conservation, however, is only one aspect, and it's redundant to some degree with some other aspects. So amino acid conservation is a bit hard to calculate. DNA conservation is easier. Amino acid identity, we would predict, is a feature that will hold true across many species, because if that principle of hydrophobic amino acids at the core of a protein is the strongest predictor in our algorithm, I would guess it's the same everywhere. Whether it's true also for other Cas9s, definitely yes. So the cutting efficiency will be different, and maybe also to some extent, but probably not so much, the type of indel that is formed, because again, this is by the mammalian machinery. But the Bioscore is independent of the cutting. So that happens long after Cas9 is gone, and is a protein feature. So that will hold true for all the other Cas variants as well. And in fact, we are looking into designing libraries for others.
Okay. Maybe a slightly related question from Russell Das. He's asking, can he compute the VBC score locally? And I guess that might be because he'd want to bring in different genome builds or work.
So I would forward that question to Georg, actually, because he put a lot of files on GitHub, but I'm not a computer geek, so maybe he can tell us how much is available there, please.
Yes, hello from my side as well. So for the computation to actually calculate the VBC score, I put there a script that should be quite straightforward to use if you have features already calculated. So if you know the exon size, the amino acids around the cut site, you can all plug this in. It's a Python script, and it will calculate the VBC score on target. For the derivation of the features, this can be a little bit more tricky, and we have so this is this does not just run locally as a like plug and press or like as a very easy application, but the VBC score is easy to calculate from feature lists. Yes.
So with feature lists, Georg, you're referring to the amino acid conservation or the amino acid
Yes.
identity at the different PAM sites.
Exactly, and if there's like a particular feature that is hard to derive because you're looking at a species where the amino acid conservation is not mapped yet, then you can also just leave it out, and it will still work. It will just apply a median to all guides that will not affect the relative guides to each other. Yes.
Okay, thank you, Georg. A question that I'm going to expand on a little bit. So, question from an anonymous attendee just asking, are we close to a one guide per gene genome-wide library? And I guess I just want to expand on that and say, does the improvement in this score sort of alter how we, or the current best practices for doing a genome-wide CRISPR screen? So, for example, the number of guides per gene or so on, or would you still advocate for continuing as people have done in the past?
Yeah, that's a discussion that we have in the lab a lot. Should we go down with the number of guides? I can tell you that with the new libraries we cloned, we chose to go with 5 guides per gene again, simply because we don't want to use the VBC score to make our experiments smaller, but rather to make our experiments better. So if we need to go smaller for whatever reason, we use sub-libraries. You know, we have libraries for all drug genes or druggable genes or surface receptors or you name it. And that, again, is very easy to implement when cloning guide libraries. But we would rather go for 5 guides anyway and then get a very good and solid result for each of those genes. Because, again, we want to go down in the list and also basically looking into the gray zone and pulling things out there. And we've discussed off-targets before. If you have 5 guides and you see a phenotype with 3 guides, then off-target is much less of a concern than going with 2 guides and one shows a phenotype and the other one doesn't.
Understood. A question from Jeroen asking whether this algorithm is applicable for alternative types of CRISPR screening. So obviously the focus is on knockout here, and you have your conservation of the amino acids potentially resulting in ablation of gene function. But is there a way that could be applied to different types of CRISPR screen, or do you think the focus here really is?
So I'm not sure if I understand the question right, but so different type of CRISPR screen. If you think of, for example, of RNA sequencing, single cell RNA sequencing as a readout or something, then my answer would definitely be yes. In fact, I think particularly in the more challenging screens where we are sort of working at the edge of detectability, there an improved guide RNA score will make a huge difference. Finding the ribosome, no, we don't need the VBC score because everybody finds it anyway. But if you want to do a imaging-based screen or something, we can't allow for too many failing guides because it's just too expensive and so on. So yes, I think for the more challenging screens, that's where the biggest difference will be made.
For something like CRISPR-I or CRISPR-A, where you're building a library for both.
Okay, thanks for the clarification. So this score is an amino acid score mostly, or a Bioscore. So for CRISPR-I and CRISPR-A, it's mostly about the spacing of the guide relative to the transcriptional start site and so on. And so no, for that, this would not be the way to go. The cutting, so the first part, the guide RNA efficiency would probably apply, but that's not making a big difference. So it's the biggest difference is the Bioscore, and that is of course not in CRISPR-I or CRISPR-A because that's a transcriptional perturbation.
Understood. A couple of people asked about the possibility of using dual guides to increase efficiency, or 2 guides per gene. Do you have any opinion on that approach?
Yeah, we don't do it at the moment. I know of several people doing it and seeing good results with that. We don't do it because, of course, 2 guides will also duplicate all the problems with off targets. And in addition to that, the cloning gets much more challenging, especially if you want to pair it in a meaningful way. It can be done, is being done. We don't do it. again, if I go back to the data I showed in one of the earliest slides, where we see a median depletion of guides targeting core essential genes of 95% depletion, that means that even with a single guide in the median of those guides, and with our old score, because these screens were done before we derived the VBC score, we already knocked out gene function in 95% of the cells. So I don't think it's necessary to go with 2 guides. Rather, it is necessary to make sure that that dramatic effect of the knockout generation is created everywhere. So on every protein.
Yeah, and I wonder if the possibility with respect to dual guides here is not 2 guides for the same gene, but to run much more robust, sort of multiplexed dual guide screens as well.
So sorry, yes. So again, multiplex screening. So for example, paralogs, definitely again yes, because there you want to make sure that you don't have too many failing guides, right? So if you can minimize the number of failing guides, you don't need as many pairs. You don't have to do A with B and or AA, BB, AB, A. If you can reduce it to 2 guides there, then of course you do yourself a huge favor. So yes, for these challenging screens, I think that's where it makes the difference for sure.
Brilliant. Okay, well, I think we've probably taken about 5 to 10 minutes worth of questions there, so I'm going to draw things to a close. Just a reminder to our audience that if you have time quickly at the end of this, there'll be a short questionnaire that you can fill in just to give us your feedback. And for those of you that want to follow up with either Twist or Ulrich, I'd encourage you to do so. Ulrich, thank you so much for a fascinating presentation. I'm super excited to see how the field continues to move forward. And thank you to our audience for joining us today. And we look forward to seeing you all at the next one. Thank you very much.
Thank you very much. Take care, everyone. Bye-bye.